Tonight what I want to do is basically roam freely. It's my final lecture of this year, which means I get the privilege of saying all sorts of stuff which might not be true, and it will be many months before I see you again, so you'll hopefully have forgotten. And what I want to do is talk about the future and where I think microbes might be going. And whenever you talk about the future, I always think of this quote from Niels Bohr, the Nobel Prize-winning physicist, who pointed out that prediction is very difficult, especially if you try to do it about the future, which I totally agree with. But the great thing about thinking about the future is as long as you do it far enough in the future, then I won't be around for you to tell me I was wrong. And so I'm going to try tonight to talk a bit about some stuff that you can hold me to account for because it's relatively near future, and some other stuff that is quite far in the future may not be true at all, and I won't know about it because I won't be here, and I guess most of us won't, so you can't correct me when I got it wrong. So I'm going to focus really on talking about a couple of kind of distance timelines around the future, and in true Star Trek fashion, I've focused on things that will come in the next sort of 25 years, I think. So those are the ones you can hold me to account for, and then ones that are beyond that are quite far in the future, very speculative, but you know, it's fun to speculate, isn't it? At least for me. If you don't like the speculation, you can quietly exit at the back or fall asleep. And what I want to do really is think about this in kind of three spheres of influence for humans, for we as humans, places where microbes are likely to play an even more important role as we go forwards for the next, say, 100 years or so. And broadly speaking, that's medicines, it's foods, and it's kind of material. So that's the lay of the land. And at the end, I am probably going to depress us all by telling us some of the things that we don't want microbes to do, but they probably will do anyway. But let's start upbeat. Let's think a little bit about medicines and microbes in medicine. And we all know, hopefully, that microbes are obviously a big problem for medicine. Many of our health issues are related to medical infections, bacterial, virus, whatever. We've all experienced just how devastating a major microbial infection can be over the last few years, and they're big problems. There are also, though, potentially big solutions for medical issues, and so this hopefully is very familiar to you. This is an early advert for the wonder drug penicillin, which, as it says in the tin, saves soldiers' lives and indeed non-soldiers too. And I suspect many of us in this room are actually alive today as a direct result of penicillin, either because we took it to treat an infection or because our parents or grandparents did, and therefore they survived and ultimately led to us. Penicillin, of course, is a microbial product. It is something that a fungus produces to kill bacteria, and as a result of that, we have harnessed that drug to treat ourselves. In an earlier lecture, I talked about many of the other antibiotics, of which the vast majority still were discovered as microbial products. So this is a medicine that microbes make to help us deal with health issues. They, of course, don't do it to help us. They're doing it for their own purposes, to fight off microbial competitors, but we've harnessed that power for our own advantage. You can also think about the benefit of microbes in a predictive way, in a way to prevent infection in the first place, through vaccination. Hopefully all of us in this room have had many vaccines, of which a very significant proportion are essentially microbes being injected into your arm or sometimes swallowed. So this picture here is an electron micrograph of the polio virus. And for those of you who've had a polio immunization, which hopefully is most of us, what you have had is a live, weakened, what we call attenuated strain of polio itself. And lots of vaccines that we still use today are essentially weakened versions of the thing you're trying to protect against. And the theory there is because if you give the body a weakened version of, for example, polio, the immune system learns to recognize all the patterns on that polio virus. It builds up immunological memory. And then when you get the real thing, the full-flavored, potentially lethal virus, it's not causing you a problem because your immune system is prepared for it. So this idea of using attenuated vaccines has been around actually for close to 300 years, because this is the original principle behind smallpox vaccination, when, of course, Edward Jenner used a very closely related infection, cowpox, and gave cowpox to a very young boy. These days, I think you would not get ethical permission to inoculate an 8-year-old boy without his consent, but nonetheless, he did this and demonstrated that exposing to a live, weakened version of a virus will protect you against the virulent one. So all of us have already been exposed to the potential benefits of microbes in protecting against other infections. But more recently, we have started to see the kind of a glimpse into the ultimate power that one can do with this, because we have moved beyond this fairly basic approach to some really advanced science. And very many of us in the room will have experienced this over the last 3 years, because if you've had the Oxford vaccine against COVID-19, SARS-CoV-2, what you have received is a microbial vaccine. But it is not a weakened form of COVID that you've received. What you have received is an engineered virus. It's actually this virus you see here. This is a chimpanzee adenovirus. So it's not a human disease. It's a chimpanzee virus that for many years has been worked on as what's called a viral vector. And the great thing about viruses is, firstly, they have a very simple genome generally, much, much simpler than, certainly than a human, but even than a bacterium, which means they're relatively easy to genetically engineer. You can move genes in, you can take genes out. So it's not too difficult to take a virus and remove genes, for example, that cause it to be very virulent, and to put in genes that you might want in there. Secondly, almost all viruses are exceptionally good at getting in and out of host cells because that's their entire lifespan. When you get, for example, a respiratory tract infection, so a common cold virus, that virus is fusing with the cells in your lung, in your upper airway, entering those cells and hijacking those cells for its own benefit. It's really good at getting into cells. So what viral vector vaccines do is they take that ability of a virus that does not cause disease in humans, like a chimpanzee one, and you put into that virus genes that are from the thing you want to protect against. So in our case, SARS-CoV-2, you put in a gene, and now what happens is this chimpanzee virus gets into us through injection, it enters our cells, and it starts producing the protein that we have put into it through genetic engineering. In this case, a COVID-19 protein. The presence of that foreign protein triggers your immune system to mount an immune response. It recognizes the protein, and then later on, when it sees the same protein, but now on a real, fully flavored SARS-CoV-2, you have some level of immunity to it. So this is an engineered virus. You're not giving someone weakened COVID-19. You're giving them a different virus that is using some genes to mimic that. Why am I dwelling on this? I'm dwelling on this because I think we have only just started to scratch the surface of this as a potential for future medicines. Because the great thing about this technology is essentially plug and play. We have harnessed it for COVID-19, but actually it was being developed pre-COVID-19 for other diseases, and now is in clinical trials for a whole range of diseases, from things like Nipah virus on the one hand, through to very extensive global problems like malaria. And so this kind of microbial plug-and-play technology, I think, offers huge potential, huge advantages for human healthcare, not just with this specific virus, but as a principle with many other viruses. And you can start to think, for example, about harnessing viruses that are particularly good at getting into tissues you might want to mount a strong immune response in, like the lung or the genital tract. And so you can start to get a very precise type of vaccine technology by harnessing microbes for that benefit. So let's start with a little bit of wild speculation on this one then. So in my lifetime, I hope my lifetime, I mean you never know, but let's hope that I've got a few years ahead of me yet. In my lifetime, I think we will see this technology harnessed for a whole bunch of immediate healthcare issues. In particular, for example, it is highly adaptable. You can engineer and produce the virus at scale rapidly, as we have seen, which means it's particularly good for infections that are very variable. And you might be aware, for example, if you already received the flu vaccine, that you need to go back every year to get a new flu vaccine, because the flu is one of those viruses that evolves very rapidly. And so you always need a new vaccine to be ready for the new strain. At the moment, we do that by a process of sort of extensive modelling and lucky guessing, basically, by looking, for instance, at what flu strain is circulating in Australia in their winter, and therefore assuming that 6 months later it will be here for our winter, and then doing some very rapid scale-up to create flu vaccines, much of which is still done using essentially immunisation of eggs and harvesting virus from eggs. It's a slow process, there's a capacity issue, and that's one of the reasons why we're not really able to respond rapidly to flu. But what about if you could sequence your flu and immediately start putting those genes into a viral vector, and you'd be ready long before that flu virus came? Or indeed many other infections that mutate, that evolve on a very rapid basis. It also offers the opportunity to engineer in other things to those vaccines beyond just the protective effect you're looking for. So, for instance, we may be able to combine the gene that you're wanting to mount a response to, so your virus of infection, with genes that do other things, for example, that are particularly good at stimulating an immune response in the upper airway. So you're generating a vaccine that is highly protective for stuff you're breathing in and out, rather than one that is sort of waiting for the infection to spread into deeper tissues. And really excitingly, I think, because this is very genetically malleable, you might be able to come up with really, really highly tailored vaccines. So at the moment, for instance, it's quite challenging to come up with vaccines for people who are immunocompromised. So young infants, people with HIV, or indeed people who are relatively old. And that's because we typically don't mount a strong immune response if you're immunocompromised. But here you have a technology where you could engineer other aspects to trigger that immune response in those individual people. You might even begin to engineer vaccines that are better for particular people. We know that the immune response, for instance, differs slightly between men and women, between different ethnic groups. And wouldn't it be great if we could come up with a vaccine that was absolutely optimal for the individual person receiving it, rather than just the standard vaccine that everyone gets in the world? And I think this is a real area for kind of exciting future development. This, I think, is all achievable essentially now, within the next few years, and I hope not to be hauled back and told that I'm completely wrong on all this in 10 years' time. But what about if we speculate a bit more widely and go further forwards? What else might we be able to do with this technology? Well, here are my starter for two ideas that are longer away and may never come to fruition, but I think might be at least potentially achievable with this kind of technology. So what about, instead of waiting to see what the next infection is and then having to respond to it, what about if we could create a preemptive vaccine library? So the way that vaccines work is they stimulate your immune response typically with a protein from the thing you're trying to protect against: bacteria, fungus, virus, whatever. What the immune system sees is a little fragment of proteins, typically in the region of sort of 10 to 20 amino acids, so building blocks long. It's a little piece of protein that it sees. And it's long enough to be kind of unique to that protein. So when you've seen that, your immune system realizes, if I see this again, it's probably associated with that pathogen. Because it's seeing a piece of protein, the total theoretical number of options that the immune system could see is finite. It's a really big number. It's up in the trillions, tens, hundreds of trillions, but it is not infinite. And so theoretically, with very high throughput technology, you might be able to create a library of trillions of different vaccines ready to go for any individual protein that you have. And then, if you're in, somewhere at the far ends of the world, on holiday, you come down with a mysterious infection, your local doctor may be able to identify that and diagnose it using DNA sequencing technology, which is already common, upload that sequence, and within minutes, people may be able to fish out from your library a matching vaccine, start to prepare it, be ready, just in case it turns out that you are the so-called index case of the next pandemic, and nip it in the bud. Maybe a bit altruistic, but I think that's quite an exciting prospect. Or what about this one? So one of the things we do now when we generate a viral vaccine, so one where you're putting a virus into people, is we deliberately prevent that virus from spreading because you don't want to give someone an infection. There are really, really good reasons for doing that. However, I can envisage a possibility where you might actually want a viral vaccine that spreads. If you retained that ability to spread, you could vaccinate someone, the first person, with, for example, a vaccine against COVID-19. They would go out, they would start coughing out that viral vaccine and immunize the people in their family, the people they meet on the tube, the people they go on holiday with, and the vaccine would spread through the population. There are, and I really need to stress that, there are huge ethical and safety considerations here. That virus might mutate. You might suddenly realize instead of curing everyone, you've given them the next big plague. And very, very importantly, how would you deal with things like informed consent? If you don't want to be vaccinated for whatever reason, how will you deal with it if the person next to you on the train is coughing the vaccine over you and you become vaccinated anyway? What about a vaccine that's not suitable for everyone? How would you control it? And most importantly, how would you withdraw it if it turns out later on that there's some unforeseen safety consequence? However, I do think there's a potential here for tackling many of the world's biggest problems. And actually, this approach is already being thought about a little bit in terms of animal vaccination, particularly in populations where they are very out of control. So if you think about things like a feral rabbit or a cat population somewhere like Australia, there's a lot of interest in measures that might, for instance, sterilize those animals through viral spread, spontaneous viral spread. So you could imagine a scenario, those of you who recall, for instance, the foot and mouth epidemic in this country a few years ago, you could imagine a scenario where a self-spreading protective vaccine against foot and mouth might have actually been a sensible thing to try and use at that stage to prevent the massive economic and welfare implications of that pandemic. So I think absolutely not trivial to consider, but something that is certainly going to be worth watching over the next, say, 10 or 20 years. So that's enough on vaccination. What else might we do with healthcare and medicines? Well, one of the really exciting prospects, I think, is what if we could harness some of the abilities of microbes to turn them to our advantage in treating disease also? And the idea of having kind of microscopic microsurgeons in your body. Why do I say that? Well, for those of you who've seen some of these lectures earlier, you'll know that bacteria in particular have a wide range of remarkable abilities. So, for instance, many bacteria produce on their surface this structure you can see here, okay, which is essentially a tiny injection needle. Bacteria use this to inject things either into host cells, like human cells. So, for instance, if you get many diarrheal diseases, the reason you're getting diarrhea is because the bacteria causing that diarrhea in your intestine is injecting into your intestinal cells a toxin that is killing your intestinal cells and causing this profuse diarrhea. Some bacteria use the same system to inject toxins into each other. So if they're fighting for competition in the soil, for instance, they are essentially kind of jousting on a microscopic scale with these injection needles and injecting each other with toxins and trying to kill each other to get rid of the competition. So they can inject stuff, which is quite important for medicine. Many bacteria are also motile. They swim really, really fast. This is Rhodobacter, which you can see here zooming around the screen, essentially at the equivalent of, I think I said this last time, the equivalent of completing the London Marathon in about 8 or 9 minutes. This is an extremely fast moving bacterium. So they move, they can inject, and the really neat thing is their movement is not random typically. It can be directional using a process called chemotaxis. So bacteria swim either towards things they want, for example, sugar or nutrients, or away from things that are harmful to them, like in some cases antibiotics. And you can see that here. This image in front of you is a microscopic grid. You can just about see the squares, I think. So this is imagine a tiny network of pipes on a microscopic scale into which bacteria have been placed. And within this network, what you will see is that we can add an attractive molecule that the bacteria want to swim towards, first into one corner, and they will swim up here, and then into the bottom corner, and you'll see in a second they reverse and all go the other way. And what you will see is that this is both fast and very, very accurate, actually. Those bacteria know which way to go. So here we are then, with a system where you have a microscopic machine that can swim very fast, it can go where you want it to go, and it can inject stuff. And this, to me, sounds like the starter for 10 for a really exciting prospect for medicine. Because you might be able to harness these individual bacteria to, rather than cause disease, help cure disease. What if you could turn these into little guided kind of cruise missiles zipping around your body to do whatever it is that we're trying to achieve in terms of healthcare? So what might those things be that you could engineer bacteria to do? Well, one possibility is you might be able to get bacteria to kill other bacteria. So if you have a bacterial infection, that's especially an infection that is somewhere difficult to treat, like in a deep tissue, perhaps you could use other bacteria to get rid of it for you. And in fact, there is already some work underway on that process. So this image you see in front of you, this is a bacterium called Delovibrio. And this naturally, actually, is a bacterium that preys on other bacteria. It swims around hunting them down, and when it finds a bacteria in a particularly disgusting fashion, it drills a hole in the outside of the bacteria, penetrates in, and then divides within its host bacteria and eventually explodes the host and escapes. which is kind of gross and cool at the same time. But potentially very exciting, because this is a bacteria that's harmless to humans, very harmful to other bacteria. So if we could find a way to make this target, for instance, I don't know, a strain of E. coli that's causing you diarrhea, or a strain of Staphylococcus that's causing sepsis, you might be able to give people this and cure the disease. Or what about if we could engineer it even more accurately to deliver, for example, a very specific antibacterial, not to the whole body, but just to the individual bacteria trying to kill? Or potentially you could engineer bacteria and other microbes not just to kill other bacteria, but to help deal with some of our biggest healthcare issues. So what if you could turn these into highly targeted chemotherapy agents for cancer? One of the really big problems with cancer, as you'll be aware, is removing cancers is tricky, especially many of the ones that are in deep tissue. So for something like a brain tumour, very difficult to remove all the tumorous tissue without damage. Often they're in places where you can't get to surgically, so inside bone, for instance. And very often what you have is you remove the primary tumour, but you have these smaller metastatic tumours all over the body that are very difficult to get to, and you essentially cannot remove surgically. But you wouldn't need to if you could engineer a bacteria to sniff them out and kill them. So, for instance, a bacteria that was chemotactic towards a tumour marker and then use that injection system to inject a toxin that kills mammalian cells could be used as a highly precise way to hunt down and remove tumours, perhaps in conjunction with classical kind of radiotherapy or surgery. And I think this is a very exciting opportunity. And actually a big step forward has come just in the last few weeks. I was already preparing this lecture and felt then very smug that at least one of my predictions was vaguely on target, because a group has recently published that you can re-engineer that injection apparatus of a bacteria to deliver not bacterial proteins, but human proteins. And that is really a first step, I think, to starting to use those bacterial injection systems to deliver things that we want them to do, rather than things that the bacteria wants to inject itself. So, that's medicine. I said at the beginning that I'm going to try and cover three big areas, and so the second one I want to think about is food. Those of you who've been coming to these more frequently know that food is a subject close to my heart, probably many of our hearts. And I like to say that food is the most important topic you can ever cover in biology, because it's almost the only thing that every human being on this planet does. Right? If you don't eat, you're not here. And so food is really, really important. So microbes are already fundamentally important for food, and I think we all know that. So if you've eaten bread or cheese or yogurt or beer, or even some things like coffee or chocolate, all of those foods that you might have consumed today were only made possible by the action of microbes, right? Yeast make bread rise, various bacterial species make cheeses or yogurt, for example, so on and so forth. So microbes have been part of human food for thousands of years. More recently, though, we have started to use microbes directly as food. And I suspect many of us in this room, certainly I have, consumed very regularly a microbe. So this is a mycoprotein-based meat substitute. And mycoprotein is, as the name suggests, a microbial product. It is filaments from a fungus that are woven together essentially to form a texture very like meat. And mycoprotein is, I guess, the first large-scale human food that is created solely by microbes. But actually, this is not a very new invention. In fact, back now more than 40 years ago, people were already exploring exploiting, I should say, the possibilities of microbes for large-scale food production. And those of you old enough to reach your mind back to the 1980s might recall this product, Protein. Not a human food, but an animal livestock food, a very high protein-based food that was added to many livestock supplements for sheep and cattle, for example. And this product was created from a bacterium. And the bacterium was particularly remarkable in that what it uses as its energy source is methanol. And back at the time, back in the 1980s, methanol was a byproduct, really, of the hydrocarbon industry, so the petrol industry. It was going to waste. And this bacteria naturally uses methanol as its building block to create cells. Those cells are extremely high in protein. And so what you can do is grow this bacteria in a vat of methanol, essentially, and then harvest it, pellet it, and turn it into an extremely high protein food for livestock. And actually, this is right back in the 1980s. The UK was kind of a leader globally in this, and this is the plant that made Protein at Billingham. At the time, the largest fermenter plant in the world producing this. And essentially, the only reason that it ended up not taking off, well, it did take off, but it ended up closing and the plant being demolished, was the global market changed. Methanol became much more expensive, animal feed dropped in price as the agricultural revolution took place, and the economics didn't pan out. But there is now renewed interest in this kind of approach, using individual single-celled bacteria, fungi, whatever, to produce food, both for animals and for ourselves. So how might we be able to do that? What kind of activities and microbes might help in that process? And I personally think that there is just a vast universe out there that we have not yet tapped up, where we may be able to do really, really exciting things. And why do I think that? I think that because I've been harping on earlier in this series about that really exciting, underexplored zoo that lives in all of our intestines. So the human gut microbiome, as I've said before, has more individual bacteria cells in it than you have human cells in your body. So you are, to a first approximation, far more bacterial than you are human. And those bacteria and fungi and viruses have a whole bunch of biochemical abilities that we have no real understanding of yet. We know that they're there, though, for very, very straightforward reasons. And so let me give you one straightforward reason. You will hopefully all be familiar with the idea of carbohydrate. When you get your food, you'll see carbohydrates listed on the ingredient package. Carbohydrate, really important energy source in our diets. Humans can only digest, we only have the genes in our own genome to digest 3 types of carbohydrate. We can digest sugar, sucrose, essentially, the stuff you have in your tea or coffee, or your donuts. We can digest lactose, milk sugar, that's the carbohydrate that's in milk, but also obviously cheeses and other dairy products. And we can digest starch, i.e. potatoes, crisps, all those kind of things. There are, however, lots more carbohydrates out there in the natural world and in the plant world, and we can't digest any of them. But we still gain energy from them because the bacteria in our guts do the digestion for us. They break down those products into molecules we can use, or sometimes they convert them into these kind of products that we then break down. And for probably the last almost 100 years, we have been sort of almost unwittingly exploiting this ability for a whole bunch of human behaviors. So there is a group of enzymes called cazymes, or glycoenzymes. These are enzymes that break down carbohydrates, okay? And we have explored and exploited them from microbes for, as I say, about 100 years. So they are used in all sorts of products that you eat or drink or use every day. So for instance, fruit juice or baking often uses these microbial enzymes. The animal feed industry, or even things like the paper industry, use these enzymes to make paper soft or smooth. And in lots and lots of home care products, you can find these kind of enzymes. And so, for example, if you're kind of my age or above, you might recall being a child. In fact, who recalls being a child, being told by your mum or dad, don't get your chocolate on that, you'll never get the stains out. Does that ring any bells? Yes? Okay. Do you hear that now? No. Now we have modern washing powders that get rid of chocolate stains brilliantly. And the reason they do that is because someone discovered an enzyme that's really good at breaking down chocolate from one of these microbes, and it's now in your washing powder, so when you put your chocolate stain in, the chocolate miraculously disappears. Or my favourite example, that's not a breakdown, that's a construction actually, is that a microbial enzyme has been used to produce a polysaccharide called cyclodextrin, and that is the compound that goes into these air fresheners that remove fragrance. So if you know, like Febreze, for example, where it says removes all horrible smells. And the reason it does that is because there is a carbohydrate in there that is really good at capturing smell molecules, a microbial carbohydrate. And so you spray it around the room, and this little microbial product sucks up your bad smells and makes your air smell fresh. I'm not sure that's actually a human critical discovery, but nonetheless, probably beneficial to many of us in our daily life. So these enzymes are used lots and lots already in our daily life. But what of the future? What might we be able to do with these that is really exciting? I think there's lots of potential here, and we are starting to discover that some of this is already happening. So, hands up if you ever eat seaweed. Good, that's very good. Those of you who watch online can't see the number of hands. There's quite a lot of hands here. This is a big component in lots of diets, particularly for things like sushi. If you are a kind of poor Western European like me, you're basically really rubbish at digesting seaweed. It's full of polysaccharides, of carbohydrates that you cannot digest, and that's a bit of a shame. If, however, you have grown up in a culture where seaweed has been a large part of your diet for a long time, like Japan, the odds are on that you actually have microbes in your gut that are really good at degrading those carbohydrates from seaweed. People in Western Europe don't, people in Japan do. The reason for that is because those microbes have acquired those genes. They're not actually from that original bacterium. They have acquired those genes for carbohydrate breakdown from marine bacteria that actually in their natural habitat kind of predate on and degrade seaweeds. So we have recently discovered this remarkable kind of symbiosis, if you like, where humans carry a bacteria, and that bacteria breaks down carbohydrate because it's already borrowed genes over evolutionary time for another bacteria that breaks down seaweed. So there is a living example of how we might be able to start engineering diets and microbiomes to help us eat interesting new foods, and that's already happened spontaneously. So what about if we could start to engineer that ourselves and create interesting microbial foods? And I think there's a huge interest in that, and one of the nicest examples I've heard about just in the last 12 months or so is from Wendy Russell's group up in Aberdeen. Wendy is very interested in creating protein-rich foods. We need a lot of protein, and particularly as we try to move to a future where there are fewer animals, because they're a major contributor to fewer livestock animals, I should say, to CO2 production. We're going to need plant-based protein substitutes. One way to do that is to turn all your farmland over to soya. That is not a sustainable strategy. And so what Wendy's group are looking for are more sustainable ways to create protein-rich foods. And they have hit upon this rather novel idea, which I love, of taking gorse. So gorse, if you're not familiar with it, is this very prickly plant that typically if you're out walking on the moorland, you discover when you sit down for a picnic and realise you've sat on one. But in many parts of Scotland in particular, this is essentially an invasive species. It's a native to the UK, but it spreads places where it shouldn't be. It's very difficult for things like livestock to eat. And so it's cleared and typically burnt. Not very sustainable. Turns out, though, that with the addition of some of these very clever enzymes, you can break down gorse and reconstitute it to produce a very protein-rich potential food. At the moment, that's done entirely biochemically, but I think it's very feasible that you could engineer bacteria to do this. And so we might end up with a future where not just gorse, but all sorts of interesting weeds, byproducts, etc., are broken down microbiologically and reconstructed into a food that is beneficial to us or our animals. And what about if you took this one step further? So I reckon the gorse is definitely within my kind of lifespan horizon, this one a bit further in the future. But what about if you could engineer microbes to deal with major human waste problems and turn them into food? So we have talked previously in this series about the recent discovery of this little chap here. This is Idionella, a bacterium that turns out to be rather good at breaking down PET, so PET, the plastic that is in drinks bottles and these things, the major contributor to ocean plastic waste, the stuff you see bobbing around on BBC documentaries and killing turtles. We need a way to dispose of PET. Turns out this bacteria can break it down. It breaks it down into all sorts of molecules that can be used to create other things. And so in the future, perhaps we'll have a system where either an individual bacteria is engineered to have lots of capabilities, or perhaps more likely a community of different bacteria synergize together to convert stuff like plastic waste into edible food, perhaps even for us. I think not entirely unfeasible. I guess it might take a little bit of getting used to. If you went to, Burger King and they served you with the ocean plastic waste burger, you might have to think twice. But, we eat lots of things nowadays where 100 years ago people would have said, really? So I would not write this off as a potential prospect for microbial food of the future. So my third area where I speculate that microbes might have a big impact is in manufacturing. And we already use microbes for lots of manufacturing, breaking stuff down, building stuff up, but there is a real unexplored universe out there of exciting stuff. In just the last 10 years or so, people have engineered bacteria, for example, to produce spider silk. As you don't need a spider to make a spider silk, you can use a bacterium, like this guy down here. We've engineered bacteria, in fact, harnessed their natural ability of some bacteria to produce calcium carbonate, rock, chalk essentially, and use those bacteria to make what was called self-healing concrete. So you can impregnate into concrete blocks a bacterium that lies dormant until the block starts to crack, and when the crack widens and water comes in, the bacteria reawakens and starts to produce calcium carbonate and plugs that crack in the concrete block. These are already things that we are doing, if you like, engineering bacteria to make really quite complicated materials for human benefit. We have also discovered bacteria that are essentially electrical conductors. So there are bacterial species, particularly in soil, that can collect metals from the soil around them and create these tiny kind of electrical wires between them. We don't really know why they do that. Partly it's because they are harnessing energy from the process of collecting the metal, but why it forms these filaments is a little bit unclear. But nonetheless they do. They're making these little kind of conductive filaments between them. So we also know of many, many microbes that produce very complicated polymers, so large molecular weight molecules on their surfaces, for instance, to make them waterproof, to make them very resistant to pressure, or to have particular properties, like to be highly conductive or not conductive at all. And there's one example here. This is Cryptococcus. This is a fungus. So out there in the world at the moment, we have microbes that can make things like the metal part of a wire and the insulating part of a wire. So it's quite feasible to use a combination of microbes to create an electrical wire. Now, why would you want to do that when we're actually quite good at making electrical wires now? Well, there are two reasons I can think of, and here's my wild speculation on this one for the future. We know, and I already showed you earlier, that many microbes respond and move to external stimuli. I talked about chemotaxis, when bacteria respond to chemicals, but many organisms are also phototactic. They respond to light. And in some cases, they respond very specifically to particular wavelengths of light. And in fact, there's been very nice demonstrations that you can use, for instance, different wavelengths of laser light to make some bacteria move one way and then move another way and then stop moving and do all sorts of interesting things. So here you have, in my view at least, the potential to create a biological microchip, because you can use laser patterning to tell your microbe where to go, and you can tell this one to go in one place and lay down a metal wire, and then this one to go to a different place and lay down the insulating polymer on top, and voila, what you end up with essentially is a microbial microchip. Now, right at the moment, we actually make microchips pretty well ourselves without any need for a bacteria. So I'm not sure how useful this will be, although I can imagine it might have significant economic benefits if you can really work it out, particularly because this is a very energy-intensive industry that is very unsustainable at the moment. Using a kind of biological process in the future might be a really good replacement for that. But in my view, much more exciting than this is, rather than having your bacteria on kind of big pipes in a factory, what about if you harness their ability to survive out there in the world? And you could imagine a future, for instance, where we spray this combination onto, say, a field. This combination of bacteria, they create, within the soil of that field, their own kind of little electrical network. And now, if you're a farmer with one of these 2,000-acre mega fields in the US, for example, you have an electrical conductive circuit in your soil, and you can use that circuit to tell you whether the soil is wet enough or dry enough, changes in pH, all sorts of things that can be deduced now. You've achieved this without any major engineering, without any mining to create the metals, without any of those things. It's a highly sustainable way of creating environmental sensors. And you could even take it one step further. What about if you make these microbes self-propagate and spread, and you end back up in that kind of ethical dilemma I mentioned at the beginning about where do you stop and how do you control it? Lots of things to consider, but nonetheless, the potential, I think, for microbes to create a totally new way of using materials out there in the world. So that's all really upbeat. I'm feeling really positive, and I am universally positive almost about microbes, but I wouldn't be doing my job if I didn't end in the last section by thinking a little bit about some of the negative things that microbes might do. And if any of you read a decent amount of science fiction, you will know that essentially the world always ends either with an asteroid or a microbe, right? I'm not going to talk about asteroids, let's talk about microbes. So are microbes going to end the world? Well, I think, unfortunately, it's probably relatively likely. We have seen in the last 3 or 4 years the unbelievable scary power of a pandemic. Okay, this is a map mid-pandemic of COVID-19 spread. in no way to belittle the COVID-19 pandemic, which is absolutely devastating for human society and many individuals. It was actually pretty mild as pandemics go, as global pandemics go. As of last week, I think the global death toll was something like 7 million people. That is a horrendously large number, but actually it's much, much smaller than many of the microbial pandemics that have happened in the past. So if you think about things like smallpox, for example, in the New World, so smallpox taken to the New World, to the Americas, by Europeans when they first discovered the New World, no immunity to smallpox in the native population of Americas, and as a consequence, over the next sort of 50 years or so, we estimate that 90% of Native Americans died of smallpox. Bubonic plague, Yersinia pestis, good historical accounts from the Middle Ages showing mortality over most of Europe, approaching probably something like 50%. So these are orders of magnitude more mortality than we've had in COVID-19. And so I do think it's really important that we don't sort of think to ourselves, we've COVID-19 done, dusted, pandemic over, let's not worry about that ever again, because there are many, many other organisms that can cause pandemics of greater sort of scale than that, and we really need to be very conscious. We have had a very lucky escape in some ways, and I think it's really important to bear that in mind. And there is a significant prospect, I think, of a pandemic being essentially that world-ending event. And I think really a very important thing to think about is you don't actually have to kill everyone to end the world as we know it. In fact, you don't have to kill anywhere near everyone to end the world as we know it. If we think about the Black Death in medieval Europe, the impact of the Black Death, firstly, was on massive mortality, but secondly, the economic impact lasted for a couple of hundred years because you'd lost all these fit young people who were working in farms. Cities had emptied as people ran away from the spreading plague. And so there's a massive deurbanization and a very long impact, which historians often say is essentially responsible for the Dark Ages because there was this long period of a failure of development in Europe as a result. And it doesn't even have to be a microbial pathogen of humans to achieve that. So if we think, for instance, about this chap, this is a fungus, okay, and it's the cause of a disease called rice blight. And this fungus lands on the leaf of a rice plant and produces this rather beautiful structure here, penetrates into the rice plant and kills the rice plant, Magnaportha. Magnaportha is a major pest of rice, and rice is responsible for the dominant calorie portion of 50% of the world's human population. So 1 in 2 people on the planet rely on this crop for their daily survival. Now, we have lots of tools at our disposal currently in terms of rice varieties, in terms of fungicides, in terms of agricultural treatment that keeps rice blast at bay. But if one was to have a highly virulent strain of rice blast, or indeed any major cereal pathogen, for example, a wheat pathogen, it would be pretty difficult to see how we could feed anywhere near the world's population. And so you don't have to kill people directly. Mass starvation and the civil unrest that would go with it would probably be enough to essentially end the world as we think of it. So that's really, really upbeat, isn't it? So I don't want to end completely on a really upbeat prospect of the end of the world. So let me give you a vaguely happier note to end on. And that happier note is possibly that we might all end in a bang, either a microbial bang, a mass starvation bang, or possibly that asteroid from outer space. But I am optimistic that even if we're not around, there will still be some microbes around to enjoy it. And that is because microbes are incredibly good at surviving all the things that we are really rubbish at surviving. So this guy here, for example, Deinococcus radiodurans, is able to withstand 1,000 times the radioactive dose that was received at 0.0 when the Hiroshima atomic bomb fell. Okay, so it decimated that whole area of Japan, wiped out essentially all multicellular organisms like us, but this guy was going quite happily. You can find this guy living, for instance, inside the reactor at Chernobyl because it's perfectly content in that high level of radiation. And then this little chap over here, Colwellia, was found at the bottom of the Mariana Trench under rock. So this is under a pressure of about 1,000 atmospheres. It's a pressure that would crush anything that humans have ever made, submarines, whatever, and yet lives quite happily and indeed divides under those pressures. And so here you have 2 organisms that would be perfectly capable of surviving that asteroid strike if it comes and eliminates humanity. And in fact, they would probably happily hitch a ride on the fragments that are ejected, zoom off into space, be unaffected by the vacuum and the radiation and everything else, and maybe land on another planet, start a whole new process. And 5 billion years from now, there'll be a guy on that planet waving his arm saying, I wonder how this all came about. Maybe they survived an asteroid strike. Let me end on that very happy note. Thank you very much for listening. And as I'm going to finish here with this famous quote from Louis Pasteur, who was always way ahead of us, who said, for those of you whose French is not good enough, it's always the microbes in the end who have the last word. Thank you very much.
Thank you very much, Professor May, for another wonderful lecture. We've got time for a couple of questions, so I'll start one off. It goes back to the medical treatment section. Could you tell us more about microbes and their potential use as nanodrug carriers?
Well, where do you want me to start and stop? I mean, so nanodrug actually is an interesting phrase, because I think that is probably one of the things where it is closest to being likely to be delivered. So for those, I talked, for example, about getting the Oxford vaccine, which was this modified virus. If you had the other vaccine, like the Pfizer vaccine, what you had was not a microbe, but a little lipid particles, a bag of fat that had your vaccine in it. And I think that's one of the areas actually where we didn't talk about, where microbes might be really exciting, because microbes are really good at producing little packages that contain things. And they're also really good at making things like drugs. So one of the big challenges with things like novel antibiotics, many of them fail because of toxicity. So you give an antibiotic to someone, it turns out that it kills the bacteria, but it kills the person too, which is not ideal. And so there's a lot of industry around what's called encapsulation, putting drugs into packages that mean that you protect the host but kill the bacteria. Bacteria are already really, really good at this. And so one possibility, I think, might be that you could engineer bacteria to take their very, very dangerous antibacterial, stick it inside, for instance, one of those lipid vesicles like we've received in vaccines, and then eject it. And then the host is safe, but the other bacteria would receive that package and die. So I think that's very feasible. So yeah, absolutely, bacterial nanomachines, here we come.
What could it possibly treat?
Well, so what could it treat? I mean, the areas abound. I mean, the most promising areas, I would say, are the ones where our current treatments are limited by toxicity. So tumour treatment is a really good example. I mean, essentially, treating a tumour is about trying to do your best to kill the tumour without killing the person who's got it. And anyone who's been unlucky enough to receive chemotherapy knows it's usually really quite unpleasant. And so that idea of hiding those toxic drugs inside a molecule could be useful. The other big area is that bacteria have obviously been exploring antibacterials far longer than we have. So they have a number of compounds that are highly specific. So for instance, there are what's called bacteriocins. So you have one strain of bacteria that might kill another very closely related strain, but not harm itself, with that level of precision that we don't currently have in antibiotics. So I could imagine a scenario in which you have very, very targeted antibiotics. Until recently, that's been of no benefit because we haven't been able to diagnose the disease with that level of precision. But now with DNA sequencing, you can tell someone not only they've got Staph aureus, but you can tell them which strain they've got and probably where it came from. So then you could lift off the shelf your highly optimal bacteria against that specific one and wipe it out. He says optimistically.
Do we have any questions? Okay. Just the mic there for you.
The magic appearing microphone.
Thank you very quickly. I'm now a journalist, and I have been doing some predictions which sadly turned out to be correct, which was that I said there would be an invasion of Ukraine. But that, of course, is very, very simple compared with what you're looking at. My question is, going back to when you started your career, what progress has been made in uses which has surprised you either on the upside or the downside? What has been exceeded your expectations and what didn't?
Well, that's it, yeah. So how good would my future prediction have been when I was 3 years old? What exceeded generally? Sure. So what exceeded? So one straight off the bat, I would say, is our ability to do genetic sequencing. So even back, I was already kind of well on the path of my career, and I remember very clearly, people sequencing, for example, one chromosome in yeast and saying, "Wow, that's a major achievement." And those of us who remember the human genome being announced, the cost of that project, billions of dollars. years of investment, thousands of people. And now, I don't know if people in the audience have done it. Put your hand up if you've sequenced your own genome. It was a couple of tentative yes, excellent. No one's quite prepared to admit it. But you can do this for, a few £100 commercially. So that's been one. And at extremely high speed. That's the remarkable thing. I mean, we routinely in my lab, sequence strains within days, which previously would have taken years. So that has been one that has absolutely exceeded my expectations, I think. A second one that has not yet happened, but I am almost guaranteed is going to, I think is going to be around computer technology generally, but AI in particular. So for those of you who haven't followed this, so protein folding is a really big challenge in biology. So proteins are these very complicated three-dimensional molecules. Their function depends completely on the 3D structure, and we have had no way to predict that. The way we've done that over most of my career is you make a crystal of the protein and you shoot electrons and stuff at it and you try and reconstruct it. 18 months ago or something, DeepMind produced software that essentially could predict the structure accurately of a protein from its sequence, something that I have. In fact, I still have the note when I was a PhD student when I speculated on this, and one of my colleagues wrote, "Nice idea, never going to happen." I should have it framed, actually. That is a remarkable thing. And now we see, that's already kind of happened. Now we are right on the cusp, I think, of artificial intelligence doing that for all sorts of things. And I am very confident that not long in the future, you'll go to your GP, and your GP won't even be a human. You'll talk to a machine, and it will say, "Oh, gosh, you've got, scarlet fever. Stay there. I'm going to send someone to come and treat you." I really think that's quite likely.
Here. Just wait for the mic. Just wait the mic.
Sorry. So if you've got, a colony of microbes doing something for you, so whether or not it's breaking down like PET or whatever, how do you regulate that so it doesn't kind of go rogue, mutate or evolve? How do you make sure it keeps doing its job?
Yeah, so there's sort of two parts to that, I think. So how do you stop your microbe from going rogue is a really good and important question, I think. So you could imagine, for instance, that PET digesting microbe, Eureka will solve all our plastic problems. Not so great if it gets out into the supermarket and suddenly all your Coca-Cola bottles have been liquidized. So I think control is a really big challenge. There are quite good ways we sort of kind of already do that. So you can engineer in things like so you can knock out genes that it requires to live out in the environment. One way that you can do that is to make it dependent on something you have to add exogenously. So you might make it dependent, for instance, on a particular amino acid. And so it only grows when you've got the amino acid, and if you withdraw it, it stops growing. That's an approach that's already used. Not foolproof, but not bad. I think a second question there is about that precision control of the process, because obviously what you don't want is a process where the degrading enzyme goes 500 times faster than the one that's making your plastic burger at the other end or something. And there you can do some quite interesting tricks. So bacteria have these processes called quorum sensing, where they can tell how many other bacteria are around and change their behavior accordingly. So you could actually fairly straightforwardly engineer a system where the degrading bacteria grows at a rate that is matched to the growth of the constructing bacteria, for example. So I think there are ways around that. But you're absolutely right that the really big question and something that needs to think about at the beginning rather than the end is that element of inadvertently unleashing the next disaster because of very good intentions.
Thanks. I'm going to follow on what he said. It's about the control. Because I'm thinking about if you were to use these microbial organisms to deal with tumors, I'm concerned that the tumor genes or genomic materials will be taken up by the microbes. And then now you get a bigger tumor than you ever thought of.
I see. That's an interesting prospect. Okay, so in terms of the genes that trigger, so there are things like oncogenes, which are genes that trigger cancer production. We actually that already does happen. So there are viruses out there that carry oncogenes and will trigger cancer. And for those of you young enough, for example, to have had the vaccination against papillomavirus, the reason for that is that papilloma itself, the virus, is not particularly harmful, but it carries these oncogenes and can trigger cervical cancer in women, for instance. And so there are viruses that trigger tumors already. So what you say is theoretically possible that you could get these genes picked up. I don't think in the context of tumour treatment that it's any more likely than it might be spontaneously to occur. So we're all infected by viruses all the time. there is some element of them picking up and getting rid of DNA or RNA all the time. And so there is a kind of non-zero possibility that they might pick up an oncogene and become a cancer-causing virus anyway. So I don't think using them to treat a tumour makes that any more likely. But you're absolutely right that what you don't want to do, obviously, is have particularly a self-propagating treatment that then suddenly gains some ability, for instance, to hide from the immune system, and now, is gone rogue. So it comes back to that same question about control. Control is really critical here, and it does need a lot of thinking through, I think.
Sorry, I just need to ask, but how is the shelf life of all these organisms? Because normally I assume you keep them by putting in liquid nitrogen.
Yeah.
But the thing is, once you sort of infuse it into another, animal, how long will they stay in there to work?
So shelf life of microbes is incredibly variable. So let me give you two examples. HIV virus, for example. So HIV, if you take it out of the human body and stick it on a surface, is dead within minutes, which is why you can't get HIV from, sharing a cup or all those things that in the 1980s we were kind of worried about. Very, very short. At the other extreme, you've got things like, anthrax, the bacteria, anthrax spores certainly live perfectly well for 50 years, probably 100 years. And in fact, we know of microbes that are not disease-causing microbes, but nonetheless exist, that seem to have existed as a single cell probably for 10,000 years, dormant actually. That's one of the exciting prospects. If you could somehow combine the thing you want to do with that excellent long shelf life, then that idea of a viral vaccine library becomes quite feasible. But it's not trivial, absolutely.
You talked about how you use the chimpanzee virus as a viral vector for other diseases, but what if the body develops immunity against the chimpanzee virus?
Yeah, very good question, and a very important one. So that's actually true not just of the chimpanzee virus, but many, many other vaccines. And in fact, one of the big challenges of vaccine development is you don't want your body to mount an immune response against the thing that's delivering the vaccine. You want it to target whatever's inside the vaccine. So that's true also, for instance, for the liposomal vaccines, the ones that don't involve a virus, like the Pfizer vaccine. It's really important that you don't react to the packaging, if you like. And that is one of the reasons the chimpanzee virus has been chosen. That one is actually particularly good at not triggering a major immune response in humans. And that's one of the factors that goes into that early selection, is making sure that you don't mount a response. You might mount a minor response, but not enough to cause a problem. But what you really don't want to do, obviously, is immunize someone, for example, with that virus. They mount a brilliant response against COVID-19, but also a brilliant response against the chimpanzee virus, and then you can never use it again because that person, one vaccine is never enough. So there is that factor of understanding what we can do to make those very discreet. But I think that's a reasonably surmountable problem, but not trivial. Good spot.
We've probably got time for one more question.
What I wanted to ask you was that temperature conditions, if it's very cold, then the microbes become inert, don't they? Because, you find bodies found on mountains, like Everest, for instance. Mallory or Irwin was found. The body was absolutely intact for such a long time.
Yep.
So does that mean that the microbes weren't able to get at it because of the very low temperature?
I see what you mean. Yes. So broadly speaking, most microbes pretty much stop dividing below about 4 degrees, which is one of the reasons why we use fridges, and certainly when it's frozen, so you can keep things frozen for quite a long period of time. And you're absolutely right that you can reconstitute microbes from these, sort of the ice. People might have come across the Iceman, this chap who, 4,000 years ago or something, fell into a glacier and has been frozen ever since. And you can reconstitute microbes from him. And in fact, but you don't have to be frozen. So actually, people have successfully retrieved microbes from things like preserved Egyptian mummies. So temperature is really important. Generally, cold chain is good, which is one of the reasons why vaccines typically are kept on cold chain when they're distributed. And one of the big challenges in medicine is moving that to a system which doesn't require cold chain, so you could use it in countries that don't have reliable electricity, for instance. But it's not a given. So there are some microbes that actually do very badly at cold temperature and are much better at kind of warm ambient temperatures. And so that is a big factor in this. And I think one of the big challenges of applying microbes to medicine is going to be that very practical part. You can have a great idea in the lab, but if it only works at 37.2 degrees on Tuesdays, that's never going to be a feasible strategy. So you're dead right. That kind of compatibility is important. And I do think one of the big attractions there is combining different microbial features. desiccation tolerance, cold chain tolerance, stability at room temperature into a thing that you could then use to treat people. But who knows? Let's find out.
We are out of time this evening. Thank you, Professor May, and thank you very much for a fascinating series this year. Next year, you'll be coming back with a series on evolution.
Yep, exactly.
Which we are very much looking forward to, and we hope you can all join us then. Thank you again, Professor May.
Thank you very much.